Hello, everyone. My name is Leonardo da Costa. I'm from Federal University of Lavras. I'm a PhD student in Brazil. Last year, I was a visiting scholar in the LIPCA lab, and today I will present to you the results of the collaboration. So the main goal here is to predict eucalyptus families across Brazil using environmental covariates. This is important because Eucalyptus urophylla is the most planted species in Brazil. The production cycle can take from 5 to 7 years, and the breeding cycle, it's way more longer. It can take 10 to 15 years. So thinking about genetic breeding, the predictions can accelerate the progress, especially if we use the G/E. So how to predict this? So the productivity here would be wood production. So the wood production equals to genetic, environmental, and interaction between environmental and genetic effects. And we can try to model G/E by treating the environment as the result of the environmental covariates. In this case, would be rainfall, temperature, soil type, and the same can be done for the G/E. So if G/E is the interaction between genotypes and environments, and the environment here is the covariates, so G/E would be G/W. And this is the pipeline we construct together. First, we need some phenotypic data for a lot of locations. Then we need W matrix, which will be a similarity matrix. We need some models to test. We need to validate the models. We need to define the populations of environments. Then we can do the predictions, we can do the recommendation, and a lot of stuff. So first, we have our phenotypic data. So here we used 18 trials, 107 families, and one commercial clone. This is the check. These are the locations across Brazil, and they are in the production sites of eucalyptus in Brazil. The environmental data was obtained in two different databases, Soil Grids and NASA Power. The soil information in Soil Grids and climate and the current weather information, which are the growth period, were obtained in NASA Power, and we applied a filter. And in the end, what we have is 545 covariates that we use to construct this matrix here. So this matrix here shows the relation between the environments, and this will bind all the environments together, mainly for prediction. Well, we need to test if this matrix is doing a good job in prediction. So this is the full model with all effects. In this case, it's not that clever to use the full model because we have no parsimony here. So what we did was to split this model in different combinations, and we validate them in two different schemes here, the CV2 and the CV0. The CV0 is the most important because we drop the whole environment to predict this. So this is the result of the predictive ability. Here we have the two best models, the model 3 and model 5. Model 5 is the full model, and we decided to keep the model 3 because it's more parsimonious. So we only have the G, W, and G/W, and both are the only that have this effect here. So the G/W are catching some G/E, which is really interesting. Then what we need to do is to filter the environments. In this case, we didn't have a really clear TPE. So what we did here was to filter using the third quantile. So the 20% most similar environments with the 18 in the training set. And then what we can do is the prediction. And after the prediction, we can rank all the genotypes and construct the recommendation maps. In this case here, we have the check, which is a commercial clone. And here we have all the families, no check. And we can see the G/E because each family are occupying a different location, and this is like a visual G/E where each family has different advantages in different places. So why this methodology is good because we can target the best families for specific regions, and we can optimize resources by not testing all families in all locations. We can test the best candidates to in the new locations that we want to plant. Thank you the audience and the IPAF, which is a company who planted all the trials, my university, the UIUC, the Lipka Lab, my sponsor, and of course the AI farms.